Good afternoon, everyone. Thank you so much for being here. My name is Anesi from the University of Pennsylvania. I have the honor of serving as this year's chair of the American Thoracic Society section on Terrorism and Inhalation Disasters, or TID. TID is a multidisciplinary group of researchers, clinicians, and administrators, and we share a common interest in inhalation disaster preparedness and response. And we encourage anyone interested in TID section activities to reach out and attend our annual meeting during the ATS International Conference in May. So welcome today to our mid-cycle webinar called an ATS TID Conversation with BARDA and the NIH Chemical Countermeasures Research Program, the agency missions, program priorities, and funding opportunities. And I'll briefly introduce our moderators and then turn it over to them to introduce our speakers and facilitate the rest of the session. So Dr. Satya Achanta is an assistant professor of anesthesiology and a member of the Chemical Sensing Pain and Inflammation Research Laboratory at Duke University School of Medicine. He studies medical countermeasures for phosphine inhalation injuries, among other areas of interest. And Dr. Shama Ahmad is a professor of anesthesiology at the University of Alabama at Birmingham, Hearsing School of Medicine, and illustrious past chair of the TID section. And her laboratory studies mechanisms and countermeasures for toxic and inhalant-induced cardiopulmonary toxicity, among other areas of interest. And then just an additional thank you to American Thoracic Society, including Samantha Carpenter, who's here facilitating today. Very much appreciate the support. So I'll turn it over to Drs. Achanta and Ahmad to introduce our speakers and get started with the program. Thank you so much, everyone, for being here.
Thank you, Dr. Anesi, for the introductory remarks. Welcome you all to the webinar. So I'm Satya Achanta. I'm the web director of TID section. First of all, I would like to thank our speakers, Dr. Yeung and Dr. Devon Andres, for taking out time to give this presentation. So first, I would like to start with introducing Dr. Yeung. Dr. Yeung is currently the acting chief of the Biodefense Research and Countermeasures branch and acting director of the Chemical Countermeasures Research Program at the National Institute of Allergy and Infectious Diseases. In these roles, Dr. Yeung is responsible for the execution of the CCRP's budget of $52 million per year, that including a portfolio of research of in the areas of basic and translational research administered by various partners across the NIH. He works closely with and on behalf of leadership at the NIAID and the NIH Office of the Director to develop and implement trans-NIH CCRP initiatives, workshops, policies, funding recommendations, communication, and collaborations. Dr. Yeung has been at the NIH for more than 17 years, and out of this, he has been serving as program officials for more than 17 years as well. So Dr. Yeung received his PhD in neuropharmacology and experimental therapeutics from the University of Maryland, Baltimore. Before joining NIH, he was a researcher at the US Army Medical Research Institute of Chemical Defense, where his work focused on developing medical countermeasures to mitigate the toxic effects of chemical warfare agents such as sulfur mustard and nerve agents. He has over 2 decades of experience in chemical medical countermeasure discovery and development, and he has authored over 40 original research, mini reviews, book chapters, and commentaries in the field. Thank you again, Dr. Yeung, for your leadership. We look forward to your presentation. Now I would like to request my co-moderator, Dr. Shama Ahmad, to introduce Dr. Devon Andres from BARDA.
Hello, everyone. I'm Shama Ahmad, and I have been associated previously and currently as a member of TID. Please join me in welcoming today's another outstanding speaker, Dr. Devon Andres. She is a distinguished biologist and currently serves as a project officer within the Chemical Medical Countermeasures branch of the CBRN, which is the Chemical, Biological, Radiological, and Nuclear Threats Division of BARDA. Before joining BARDA, she worked as a scientist at the US Army Medical Research Institute of Chemical Defense, the USAMRICD. Dr. Andres earned her PhD in biomedical sciences from Oakland University, where her studies focused on health and environmental issues. She also holds a graduate certificate in forensic toxicology from University of Florida And as a certified toxicologist by the American Board of Toxicology, she has over 2 decades of experience in both in vitro and in vivo research, specializing in inhalational toxicology, immunotoxicology, neurotoxicology, cutaneous toxicology, and toxins of biological origin. Her research focuses on understanding toxicological mechanisms and developing targeted therapeutics for toxic agents. And she also has a strong background in product management and the development of related cutting-edge technologies. She will speak about BARDA missions, program priorities, and funding opportunities. Once again, please welcome Dr. Yeung and Andres to the platform. Thank you.
Dr. Yeung, you can start sharing the screen. Wonderful.
Right. Yeah. Thank you, Satya. Thank you, Shama, for the invitation to speak about the NIH Countermeasures research program to the group. Without further ado, I will just go ahead and get started then. Let's see. Okay. Yeah. So real quickly, what I'm going to do, I'm going to give a brief background, historical breakdown of the CCRP, where we are at right now and where we're going and how we're going to go there with the various funding opportunities that we currently have active on the street right now. So with that, I'll just start with that. The CCRP, the goal is very simple. As you can see, we're here to support discovery, research, and early development of MCMs to advance the nation's medical and public health preparedness for response to and recovery from disasters and public health chemical emergency. So this is a perfect venue to speak with the TID group because our focus is, as the statement says, to develop MCMs for industrial accidents and terrorism. So, as you can see on the screen, the first panel and the third panel are industrial accident, train derailment, industrial acts, and industrial accidents. And then the panel 2 and 4, it's large-scale mass casualty, deliberate release of chemicals. 2002 Moscow theater in the second panel, and then release of chemical warfare, in this case, sulfur mustard, in the fourth panel during the Syrian civil war. And if you want to learn more about the strategic agenda for the CCRP, the link is right there. You should take you to it. So real quickly, as a brief background, the CCRP is the third arm of the overall NIH Biodefense Initiative that is administered by NIAID. It complements the Biodefense or Biological Emerging Infectious Diseases program that was started in 2003 by NIAID. The current director of that program is Dr. Paula Bryant. And then, two years later, in 2005, NIAID stood up the Radiation and Nuclear Countermeasures Program. And the current director, as you can see, is Andrea DiCarlo-Conan. And then finally, in 2006, the CCRP was stood up to complement and to cover the whole CBRN initiative at NIH. So the genesis of the CCRP really began in about 2003 as a result of the terrorist events of 2001 between the World Trade Center attack and then the subsequent anthrax mailing. Immediately after that, it became apparent to more apparent to the U.S. government that asymmetric attack on civilian population is a very major growing area of concern. So in 2003, after the BioBoat program has stood up and the revenue program was moving ahead, the HHS and NIH Office of Director directed Tony Fauci, the director of the NIAID at the time, to conduct a expert panel on medical chemical defense research. And the point of this was really to provide a lay of the land overview of where we are at as a nation in terms of medical countermeasures research in chemical defense, and more importantly, to identify gaps in scientific knowledge critical to developing medical countermeasures to protect the civilian population. And that's key, the civilian population, because up until 2001, before the attack, almost all of chemical defense MCM research was either conducted by or funded strictly by the Department of Defense. And with that, the Department of Defense has a very different mission where their focus is on protecting soldiers, get them, if they get injured, in this case, chemicals, how to get them back on their feet and back on the battlefield. So their focus is really on very healthy subset of the population, 25 to 40 years old, mostly male. So, so that left a lot of the general US population at a disadvantage, because obviously we have different subpopulation, pediatric, pregnant, geriatric, people with pre-existing medical conditions, and of course females, to complement the male focus of the DoD. So that's where we're trying to get the first lay of the land and identify ways for NIA, NIH to complement those DoD, Department of Defense efforts. So as part of this workshop, NIA brought in folks from academia, from industry, various state agency, poison control, and of course the DoD, and then National Academy of Science, Institute of Medicine, all the other NIH institutes, as well as the intelligence agency, CIA, DIA, to really understand where we are at and what were the gaps at the time. So with the two-day workshop, the main recommendation that came out of the meeting, as you can see, are the 8 listed right here. And it's really broken down into the need for more basic research and then apply research to identify medical countermeasures, not just new therapeutic approaches, but actually ways to repurpose therapy that are already FDA approved. So in essence, expanding the label of already licensed products. In terms of basic research side, there was a need to further understand mechanism of injury, in terms of the various organ, tissue, and cellular types after chemical exposure. And again, number 2 is what I alluded to earlier. What about the various subpopulation? How would they be affected, if, based on their pre-existing conditions? Developing animal models, in vivo and models was a real critical need of the program. And then, number 4 is to see what are, long-term, low-level, acute, chronic health effects. So if a victim survives the chemical attack, what's next? What are their long-term prognosis? So these are things that, major findings from the expert panel workshop. So with that, in 2005, NIH received its first appropriation to stand up the Chemical Countermeasure Research Program. So our program received $49.5 million in 2006. That's when we started. Fortunately, for the next 7 years after the program started, we saw a continuing decrease in the budget that Congress appropriated to the NIH. And then we saw a rise after that from 45.9 up to 52.8 this year. Again, over the course of about 18 years of the program, our budget has only gone up $3 million. So absolutely did not keep up with inflation, but so it went up, but not really. One takeaway from this graph is that even though our 2024 budget was 52.8, as many of you may have remember, it took a while for Congress to finalize the budget for this fiscal year, for fiscal year 24. At one point, we were told that to expect a 20% cut to our budget. So that would have taken us below this table. So, I just wanted to get this out there, because since there are a number of people on this call are grantees or, of the CCRP. So, and they've always asked, why haven't our budget or your budget been allowed to go up over the years? This is the driving reason why, our budget. So just take that into consideration. Anyway, so what do we do with our budget? So as you can see, again, the goal is to support early discovery research and development of MCMs. We have two strategic objectives that came out of the workshop. One is to really stimulate and sustain a broad research community, to really push for more basic research to expand the current knowledge base. Again, as I mentioned, until the CCRP came to being, most of the basic research on chemical toxicity was resided mainly within the Department of Defense funded effort. Our goal was to really grow that civilian research base. And we want to support basic research that not only expand knowledge base in terms of the toxicity of knowledge of toxicity of chemicals, but as well to address knowledge gap in terms of the acute and long-term chronic non-lethal effects, and then how these effects may differentially affect special populations. So that's one of our objectives. The second objective is a applied translational research objective, which is to apply the fundamental knowledge gained from the basic research objective towards discovering novel medical countermeasures approach, novel chemical molecular entities, helping and repurposing existing or advanced therapeutics as broad spectrum use based on shared clinical indications. So I've been talking about chemical threats for a bit now. The next question is, so exactly the who, what, when, where, how of chemicals. Who defines chemical threat? How do they do it? And what are chemical threats? Fortunately, that's not something NIH does. We're not the policy shop. We don't do the research to say what is or isn't a chemical threat or a chemical of concern, so we call it. That's actually the purview of the Department of Homeland Security, more specifically the Chemical Security Analysis Center, the CSAC, that resides within the Science and Technology Directorate. So what they do is they have this system, this approach called PANTA, which is the probabilistic analysis for national threat, hazards, and risk assessment. And they take data of known toxicity of chemicals with known toxicity, feed that into a database, and feed into whether there are known therapeutic approaches, therapeutic strategies for these chemicals. They feed in availability, ease of synthesis, access to these chemicals in the general public, and then finally they put in any intelligence that they may have received or from the intelligence community to determine whether a chemical presents a public health threat. And what comes out of this analysis is what it used to be called the Chemical Terrorism Risk Assessment, or the CITRA list, which is a list of chemicals where DHS has identified as credible public health threats. As of the last iteration of the PANTA CITRA assessment, about 200 chemicals have thus far been identified as chemicals of concern. We cannot, with 200 different chemicals, we cannot take the old one bug, one drug approach to finding therapies. So what the US government has done is group these 200 chemicals into toxidromes of interest. As you can see, this is just a quick breakdown. These toxidromes are based on primary symptoms of toxicity. Again, this is just primary. We all know chemical, depending on route of exposure, duration of exposure, can exert toxicity on different targets. In any case, these toxidromes are generally broken out into pulmonary, respiratory, or AKA pharmaceutical-based agents, vesicants, cellular respiration inhibitors, neurologicals, and others. What you see presenting the numbers are roughly, 60% of the 200 DHS chemicals of concern are considered pulmonary, under the pulmonary toxidrome. So these chemicals are, as the name implies, targets the lower and or upper respiratory tract, cause pulmonary edema, capillary leaks, pulmonary fibrosis. Some of the chemicals are chlorine, phosgene, sulfur mustard, which several of you guys on the call are already working on within the program. Synthetic opioids, nitazene, fentanyls. Not only are they the public health concern as under the opioid epidemic, they're also a DHS chemicals of concern in terms of terrorist use. And as I said, the vesicants agents, so respiration and neurological. So primary reason for the toxidrome of breaking these chemicals of threat into toxidromes, again, is to help guide the therapeutic approach, where the goal is we're working on therapies, MCMs, that treat the injury, the symptoms of injury, of intoxication, rather than the agent. So we're not looking for antidotes. We're looking for things to treat what results from the exposure. So with 200 chemicals, so many toxidromes, the NIH had to take a holistic approach where we partner with various institutes throughout the NIH. So for example, for ocular concern, we partner with the National Eye Institute so that they have the expertise to help manage and, more importantly, outreach into the research community to find ocular researchers to bring into the program. And then, as you can see, with NIDA pharmaceutical-based agents, neurological, we work with NINDS. Pulmonary, we work with NIEHS, as well as NHLBI for the program. Developmental research, NICHD is the perfect partner, and NIAMS for the dermal research. We just started a new partnership with NCATS to leverage the CTSA program, the Clinical Translational Research Program, so that we can hopefully leverage and partner basic researchers that we fund through under the other institutes with the clinical expertise and translational expertise available through NCATS. This is just a high-level overview of some of the initiatives that we have within the CCRP. I'll go into these in a little bit more detail in the next couple of slides, but as you can see, as I said, our focus is on both basic and translational research. So for basic research, understanding disease biology, early targeted identification research, we have initiatives that covers R34, which are very small planning research grants. And then we have R01s and R21s. We also now have an exposure resource and coordination core at Wake Forest Institute of Regenerative Medicine, and that's in essence to set up a mini quasi CRO facility that can partner with researchers who may not have exposure resources in place. So it's definitely a core for the community. In terms of our translational and applied research resources, we have what many of you may know as the Counteract program, and that's mostly the cooperative agreement, the U54, the U01, UG3, UH3, and the translational R21. So that's one that a lot of you are more familiar with. To complement the cooperative agreement program, NIAID has designated the MCM initiative as a high priority area of interest. So with that, we have a paragraph in the Omnibus SBIR STTR solicitation to recruit for research effort to develop medical products. And then we also have an interagency agreement with the Department of Defense and HHS BARDA. This interagency agreement, if you don't know, are just contracts between government departments to foster collaboration. Then we have a couple of research resources as well for PDRs, so preclinical development research support, as well as screening programs. Not really going to get into them for this purpose of this talk, but I can send out the slide deck and you guys can go and take a look at the web links for these. So in terms of where we are at right now in FY24, our program is at its largest it has ever been. We have a total of 91 projects funded in the program. As you can see, breaking them out to neurological, cellular, respiratory. The program is pretty much even across the board in terms of toxidromes, except for cellular respiration, which we're working on in dermal. And then you see the various funding mechanisms that we have in place to support the applied and basic research. What I'm happy to say is that as of right now in FY24, at the conclusion of FY24, our basic research portfolio is slightly larger than our applied research portfolio. And that's actually a very good thing, since we're hoping that the basic information from basic research can inform the community and drive the applied research program going forward. Just a quick representation of our current portfolio. As you can see, right now we're primarily concentrated on the eastern side of the U.S. What the hope is, with so many toxic industrial chemicals being chemicals of concern, and they're widely used in the Midwest as well, that we can tap into this scientific expertise that are out there to really grow the program and balance our portfolio even more and drive the science. Here's another representation of our current FY24 portfolio across the different toxidromes and the chemicals of concern that are currently covered. So I mentioned earlier, there are about 200 chemicals of concern identified. Our portfolio is just touching, maybe a little bit over 10% of it. There are about 30 chemicals covered here within our portfolio. So lots of the science is out there. There's lots of opportunities to come into the CCRP research program. Yep. So yeah, another way to look at our overall portfolio. It's well distributed between neurological, dermal, and ocular. But our portfolio, in terms of spanning for pulmonary, is one that we are hoping we can grow much more. And again, something that I hope can come out of this presentation to ATS and TID. And for anybody who's interested, these are the funding opportunities that are currently active right now on the street that we are soliciting research for. Just going to really quickly go over the last couple of slides where I will just focus on the 4 that are starred here. These are the more pulmonary research ones that this group may find of interest. So we've designed our program with our NOFOs, our funding opportunities, in sort of a stepwise manner, where we have our 34R01s to support basic research and then apply. I'm going to go through this really quickly, stepwise. So for basic research, we have RFA ES005 that's currently soliciting application. This is for a 3-year R01 up to $300K annual direct cost. The goal for this initiative is to support fundamental research only to characterize the acute toxic or long-term pathophysiological effects. Chemicals concerned the pulmonary chemicals that cause edema, pulmonitis, and fibrosis. And examples are the chlorine, phosgene, sulfur, and oleum. The key thing for anybody interested in this funding opportunity is absolutely no efficacy studies, no MCM candidates. This is a strictly basic research understanding mechanism of toxicity, FOA. If you have potential tool compounds to use to validate targets, that's fine, that's okay. But just don't call them MCMs, don't call it efficacy studies. These are validation studies. And we also support R01s and R21s submitted under parent announcements. So these are the standard NIH solicitations as well. For exploratory research, if you're interested, we have the translational R21 program, 2-year, $275K direct cost. This would be efficacy study, proof of concept, efficacy study, small scale. And then, to follow that up, we have the UG3, UH3, larger translational studies. The current solicitation right here, this current solicitation is actually expired, but we have a notice out that for a notice of intent to publish to renew the solicitation that should be coming up fairly soon. And as you can see, these are for 2 to 3 years, up to 350K projects. Additionally, we also support SBIR STTR submitted under the parent announcement to support exploratory applied research, translational research. And then early stage development research, same FOA. This is the second phase of the project, which is the UH3 phase, which can cover 2 to 3 years, up to 450K. Again, this is for more candidate optimization, in vivo demonstration of activity, and efficacy in more human-relevant post-exposure models. The end goal of this is to deliver at least one well-characterized delayed treatment therapeutic candidate against the actual chemical concern, not a surrogate. And similarly, we can also support this type of research through the SBIR STTR announcement. And then the mid-stage development research will be just the phase 2 SBIR project, 1 to 3 years. Again, if you're interested, you can look at the program description in here. And then finally, for advanced research and development, I'm not going to touch on this because Devon will cover the BARDA perspective right after this. So that's how it looks. Again, in our FY24 portfolio, we have 91 total projects, 14 government projects with the Department of Defense, 77 total grants and cooperative agreements, and of those, 28 were new starts. So they were brand new projects that was just funded in FY24. And more excitingly, 20 of the 28 are first-time CCRP grantees. So whopping 71% new programs, new entry into this field. Some successes with the CCRP over the years. These 8 products that have come through the CCRP grant program that have transitioned to full and BARDA advanced research and development contracts. And if you have any questions, these are the contacts to reach out to based on the research area of interest. And that is it.
Thank you, Dr. Yeung. So in the interest of time, now Dr. Devon Andres will start her presentation, and we will save all the questions to the end of the webinar. And in the meantime, if you have any questions, please post them in the Q&A, and we can compile questions based on thematic questions, and then we can present to the speakers. Thank you all. Dr. Andres, you are muted.
Yeah, that's what I realized. I didn't unmute myself. I want to say thank you, Satya, for inviting me and Dave Yeung to speak to you today. Today, I will be talking about, more of how BARDA does our chemical medical countermeasure development to enhance our community preparedness. Dave gave you a pretty good start, but I'll give you a little information on ASPR and BARDA itself. First, ASPR's mission is to assist the country in preparing for, responding to, and recovering from public health emergency and disasters. This is done by developing, stockpiling, and distributing response tools against multiple threats, sending clinical response teams to places in times of crisis, and ensuring our healthcare and public health partners have the knowledge and tools they need to navigate today's healthcare security threats. Now, BARDA is under ASPR, and our mission is to develop and procure medical countermeasures to address public health emergencies as well by utilizing partnerships with industry, academia, and other federal agencies to promote innovation and flexible authorities and providing multi-year funding and cutting-edge expertise. Now, Dave Yeung actually did a pretty good start on a little bit of what we do. First, we actually follow and look at what the NIH and the CCRP program are developing. They are one of our major, I want to say, places where we look for our pipeline. And we also follow the DoD as well as industrial partners that we know are preparing or have clinical indications that we are interested in. We do have a full development pipeline that goes from hit to lead all the way out to procurement. Most of our programs do not do basic research or early development. We mainly do most of the advanced development. That is part of our name, the Biomedical Advanced Research and Development Authority. Within the ChemMCM program, we do like most of our products to come, or candidates to come in with a clinical indication that has already had phase 1 completed, phase 2 and 3. And then we can go all the way out to procurement and into the SNS, as well as vendor management inventory, depending on what the threat is. Now, as Dave says, we do work a lot with industry partners. Throughout the years, this is a lot of our partners that BARDA themselves has worked with. And it seems to be growing all the time. Pretty soon, I think we're going to be off the page, or so small you won't be able to see anybody's logo. So now, overall, BARDA has supported many different efforts. This is outdated a little bit while I was getting cleared for public release. We've had another one. So we've had 96 US FDA approvals, licensure, and clearances. They cover devices, diagnostics, therapeutics, vaccines, different areas of threats, including Ebola, monkeypox, Marburg virus. They have antimicrobial resistance, as well as what we are interested in is chemical threats. Now, within the CBRN division, we've had 35 FDA approvals and licensures and clearances. I'm going to just show you a few that are chem-related. This is including the Meridian Cesalam, the Agenta Medical Servalon for sulfur mustard, not silver, and then the OPV nasal nalmefene spray by Indivior. Those are just a few. In the next few years, we probably have 3 or 4 more coming being approved by 2027. Okay, now the chemical MCM program ourselves, our mission is to improve health outcomes for all affected by chemical exposures. Our goal is to ensure that the medical countermeasures for chemical injury are readily and available with our end users. This involves multi-purpose drugs that are threat agnostic and broad spectrum. We look at repurposing common drugs for chemical indications, similar to what Dave was talking to you about. We build upon what they look at and then go through and try to get it through the FDA or with concept of use for the field, so we can teach our clinicians if they don't already know how to use them. We like our drugs to be as far forward as possible, specifically already with the end user, already on the EMS or ambulance whenever possible. And we do a lot of our user-focused engagement. This is because we don't want to make products that the first responders won't use. They need to be able to use it. They need to be able to know how to use it. If we make something that they ain't ready to use, then what's the purpose? So we really need to involve our end users in all of our engagements and our products. Now, a little bit about chem that's different than a lot of our BARDA agents or threats speed is the critical component of our response. Even the most advanced countermeasures fail unless present in sufficient quantities with minimal delay at the location of need. So this leads to this part. As Dave said, there are a lot of different chemical agents that are on the DHS's Chemical and Terrorism Risk Assessment list. There are over 200. We can't produce and we can't procure as many MCMs for each of those alone. Also, we don't know where or when they will happen. So we can't just stockpile them. We can't have them just willy-nilly anywhere. So we need to try to prepare them to be near where those exposures are going to occur. And no matter what the effect of a chemical exposure, we do know that its damage will occur within minutes to hours, making stockpile impractical. This is a little bit different than what you would see with biological threats, including, or even like COVID and the pandemic. You have days to weeks sometimes. Unlike with chemicals, we only have minutes to hours. So it's important that we have them as far forward as possible. So we've done some research, and one of the things that we determined and found out is that in a chemical exposure, that the first step in an incident is physical decontamination. That can remove 99% of chemicals on the skin. Very important. After that, that comes in with the medical countermeasures. So with those medical countermeasures, we like to have them be able to treat injuries that are very similar, that have characteristics similar to other clinical indications. It is our goal to demystify emergency response, where during a chemical exposure treatment within a community, it won't be confusing. No one will be just going, oh my God, what do we do? What do we do? But treat it more like a poison control type of response. And hopefully the most important question is, if the pharmacy is not open, and if it's not, can we actually use a brick to break open the window and actually get what we need? Because it is that far and more common use. High goal, but that is our goal. Now, as Dave said, we have went to the development approach of treat the injury, not the agent. And we have been doing this for quite a few years now. And to that effect, we do break down all of our threats within the 5 toxidromes, primary injury. Dave gave you a huge list, but this is what we do. Okay. Again, treat the injury, not the agent. Now, the advantages to treating the agent or the injury and not the agent is that it provides us a multifunctional pipeline for normal use of that allows the normal use of practice of emergency medicine within where clinicians treat what they see, and it's agnostic because we are not concerned so much with what caused the injury, but the injury itself. And again, it allows us to provide clinicians the ability to use drugs that they know, that they have on hand, reducing the need for stockpiling and complex deployment strategies. So, red agnostic drugs enable public health preparedness. Very important. Now, some of our BARDA user-focused successes include the recent nalmefene intranasal delivery of nalmefene in OPV. It has the potent and long-lasting reversal of opioid overdoses. We've had the first ever medical countermeasure approved in the US for treating sulfur mustard injury. This is for the thermal and radnuke burns as well. So again, it is a truly agnostic, multi-purpose countermeasure. We also have midazolam. It is commonly used for it's a commonly used benzodiazepine. In the past, we have tried to develop treatments which were approved with clinical indications to treat seizures caused by nerve agents. BARDA, in collaboration with JPO and with information provided by like the CCRP, we have developed the IM formulation of midazolam that is approved to treat all seizures. Important is a seizure is a seizure is a seizure, whether they arise from epilepsy or result from exposure to nerve agents or organophosphate pesticides. Remember that because that's an important comment I'm going to be bringing up later. We also have your emergency response resources. This is PRISM that gives you guidance on the human decontamination and processes. We also are developing a user-friendly app for CHEM, which stands for Chemical Hazards Emergency Medical Management, and that provides us information resource on medical management of these chemical incidences. Kind of like your your poison control, but on a phone or in your hands. We also have other things in the works like sublingual atropine, as I was talking about far forward as much as possible. Atropine eye drops are used all the time for your in your eye, your ophthalmologist, they dilate your eyes. Here we are looking at to be able to use sublingual atropine as if we have a mass casualty situation where mild to moderate exposures can actually use these eye drops. Just put 3 to 8 drops under your tongue and you actually be able to show protection, just like with your atropine IV infusions or auto injectors. We're also looking at respiratory stimulants. This is in response to, or similar to like what you look with respiratory depressants, nalmefene and naloxone. They direct directly with opioids. However, they're not the only ones we need to protect against. So we are looking at respiratory stimulants across the board. That way we can restore breathing and let them sleep without actually well, let's just say not everything's going to react to an opioid antagonist. So that's what we're looking for. This will also treat opioid overdoses and your mixture overdoses as well. We're also looking at improved auto injectors. We have one with a tracting needle. This is with 2-PAM. It was an oxime reactivator for nerve agents, as well as a needleless with our midazolam auto injector. So we are actually working on trying to get our midazolam / Cezalam approved in auto injectors so we can actually have them within the SNS and ready to use when we need them. And we are also looking at repurposing of the drugs to treat chemical injuries, and we have several in our pipeline. This is just a little bit more about OPB. It was BARDA's 76th approval, and it is, besides any terrorist threat, it also helps with the community and their overdose problems. It is, if you look, the fentanyl half-life is 4 hours, naloxone's half-life is 1 hour, well, nalmefene's half-life is over 10 hours. This reduces the stress and burden on our medical system and helps our first responders, especially if they need to go in or they get exposed themselves. It's also there to help bystanders and family members, police and first responders. And as it says here, is approved for people 12 years and older. Currently, we have it on contract to actually be put in for procurement. So now, there are several different pathways that you can work with BARDA, or actually how we actually get our medical countermeasures through. We have it where you could develop new drugs for clinical and chemical indications simultaneously, utilizing common symptoms like wounds, seizures, ARDS, ALI. Or you can look at new indications or route of administrations for use of available drugs as medical countermeasures. This includes label expansion or just new concepts of use, like the sublingual atropine. And this provides us with a shorter and less costly path for FDA approval. Now, this is just a little bit of our current portfolio, along with their clinical indications. Working with BARDA, as we said, we are looking at treating the symptom and the injury and not the agent. So we are asking or requiring a lot of our medical countermeasures when possible to have a clinical indication that is not related to the threat agent itself. Here are some of the examples, like with Genentech, Alteplase. We were looking at it for a mustard inhalation threat, but its clinical indication is for fibrin airway CAS. They have it so far for stroke, but we are looking forward to use it for CAS formations within the lung following a mustard exposure. Or like with Inversa, this is for an ocular. We're looking at INV, I think it should be 2. So that's a typo. It is for mustard-induced ocular injuries, but we are looking at keratoconjunctivitis, dry eye syndrome, as well as diabetic retinopathy. And if you look here, every single one of our partners, they have a product that is multi-use. For our threat with nerve agents and opioids, phosgene and chlorine, but they all have a clinical indication. This also shows you a little bit of some of our priorities. Right now, our priorities are phosgene and chlorine, respiratory toxicants. There's very little out there that we know that can be used to treat these. And it's been one of those that has been studied in early development, but has not success so far in advanced development. We're still doing some of our nerve agents and opioids, as well as our mustard, across different routes of administration or exposure. Now, if you can see, a lot of what we are doing is across the board, where we have multiple pathways to get to these treatments. We have several methods for identification, where we look at non-clinical and natural history. We have organ chips and microphysiological systems, as well as landscape analysis that includes in silico and AI and machine learning. Taking some of that information, then we can go into some screening models that we have that provide high throughput screening, either with organ chips or within our non-clinical screening models. We actually have a couple with collaborations with the CCRP that we can look at for right now. The main ones we have is sulfur mustard inhalation as well as ocular. We also have some capability within our redirect EasyBAA, where we can actually do some proof of concept studies so that we can see if we can actually put you in and be successful within a full BAA contract. I'll discuss some of this a little bit later on as we get through these. We also develop primary clinical indications at a cost share capability, where we actually support studies for your clinical indication, for your phase 1, your clinical trials, as well as your CMC and formulation efforts. We also support studies, either non-clinical or early phase clinical trials, for looking at the use is in the chemical injury. And then we do the advanced development and research, and then we also can procure and field those successful medical countermeasures. The main way we can work with people is through the BAA, or area of interest topic number 5. We have multiple different areas within that. This actually provides or requests abstracts for projects to develop these medical countermeasures to prevent or treat acute and chronic effects of chemical threats. These include opioids, chlorine, phosgene, sulfur mustard, cyanide, and nerve agents. The important things to remember is it should have a relevant chemical indication that is similar or identical to symptoms of that chemical injury. As I said, the keratoconjunctivitis conjunctivitis, if it's a sulfur-mustard ocular or something similar. It's very important. This is for making sure that after we get it through approval, the company itself is still going to be successful because you're going to have the commercial capability and business success as well. It is, it's small. Working for the government, you only get small little pieces. So we want to make sure you have those capabilities and tools to be able to be successful with, with those products that we help you develop. And the one big thing that I know that's a big difference out there than what a lot of people think is that we try to avoid specific chemical indications as well as the animal rule regulatory pathways whenever possible. It is, in our experience, it is easier to actually do a new chemical entity approval rather than in humans, rather than trying to do the animal rule. You have to do a lot of hoops on knowing what your natural history is. You have a lot of things where you got to prove to the FDA that your animal models are similar to what you would see in humans as close as possible. And sometimes that's hard to do. We've had problems now, because a lot of these chemicals like sulfur mustard or that's the main one right now that I can think of. But with that one, you have issues because there's not that many people that have been exposed to sulfur mustard. The ones that have, it was years ago, decades ago. And so a lot of times when they were treated and diagnosed at those times, it wasn't using the same technology or the same wording. And so it's hard to actually correlate between what we're seeing and diagnosing in our animals in these non-clinical studies and relating it back to the human condition. So that's why we try to get those medical countermeasure candidates that have a very similar symptoms or mechanism of action that are tied to other of those diseases and syndromes. That way we can get approved for those and then do the bridging studies, not animal rule studies, but bridging studies to be able to understand the treatment regimen, how often, when, what are the effects you need to look for, all those little important information. We are also looking at, within our BAA, we are also looking at we have a new subtopic that includes the development of human relevant models of natural history of chemical injury. As I said, we have run into problems with the FDA. No animal model is going to be as good as the human. However, we can't do that in a lot of our cases. So we do have to look at syndromes and diseases that are very similar. And I hear a lot of times that we do what we do. Hopefully we be successful in getting these medical countermeasures, but our goal is to never have to use them because, it is, I don't want to be exposed to any of these things. Let's put it that way. Now we do have the redirect program. I know I mentioned this a couple times, which stands for repurposing drugs in response to chemical threats. And the goal of our redirect program is to identify the therapeutics that could be repurposed as medical countermeasures against chemical threats to prevent injury. And then we have program supports. We have our program can support companies in collecting that critical proof of concept data for their drug and models of chemical threat injury. Basically, what this is saying is, say you have a drug for idiopathic fibrosis, lung fibrosis. That's what you're going in for with the clinical indication. You already have a lot of your toxicity data. You are in phase 1, phase 2 of your therapy. But it's like, oh, my mechanism of action should be able to help treat some of the symptoms that these chemical agents and whatever toxidrome it is. Well, the redirect program provides you a small bit of money to be able to do those proof of concept studies or reformulation studies if necessary, depending on what it is. So it is a very nice program. It's not a full BAA. It's an easy BAA. You basically do a white paper type abstract and we go from there. Now with this program, you do, as I said, small amount of money. It's 750,000. So it gives you a little but then you also get our expertise with our regulatory, our CMCE, and clinical experts. So it helps. And in this program, our holy grail, what we're hoping for is to have a readily accessible, provider familiar, safe, effective medical countermeasures that are available at hospitals, pharmacies, and within the community. Okay, I'm going to actually just put a little key in here too. I am going back a little bit. In here in our screening model, I was just going to emphasize that we do have some of our non-clinical network screening capabilities where we have collaboration with a CCRP. A lot of this is where you can, if you think your countermeasure candidate is a good fit, our toxidromes specifically, well, right now I know the mustard is the main one that we have out there. We also have some with chlorine. If you think that yours is a good candidate, we have capabilities to screen your medical countermeasure. We just put a MTA in or a medical material transfer agreement in. We do the studies. We provide you with a report, and then you can use that in any way you see fit. Hopefully, if it has a good outcome and efficacy, we will then look into seeing about getting it into our program. So that's something to consider. Now, when you do like to work with BARDA, as I mentioned earlier, we like you to be in at least a phase 2 or 3, or even have approval for a certain your clinical indication. But we also need to make sure you have your manufacturing capability, that you have met with the FDA already. We say even before, that's almost a necessity to actually get awarded at BARDA, or at least with the chem program. You need to talk to the FDA. You need to make sure you are on the right path in the regulatory strategy that will be successful for you in your compound. It is very important. So they always say meet with the FDA early and often, and it is definitely something you need to consider. So I think that's most of what I had to talk to you today. But so in summary, remember, we like to treat the symptoms strategy that brings chemical preparedness into the realm of the routine medical toxicology, emergency medicine, and pre-hospital practice. We want to try to use everyday drugs as much as possible, leveraging the physician familiarity and community accessibility. And then our program supports repurposing of approved and late-stage development drugs to treat the effects of those chemical exposures. There are many different ways you can work with us. But first, if you have any questions, reach out to myself or our, should have changed it from branch chief. It is program director, Judy Laney. We also have our link to our website here. And then we do have various areas and ways you can actually contact us. So if you have any questions, feel free to look at those sites or ask me or Judy any questions you have, and I will hand it back over to the program.
Thank you so much, Dr. Andres, for the overview of BARDA program and how it supports the researchers and medical countermeasures research. So again, I would like to take a moment to thank both Dr. Yeung and Dr. Andres for your time and for this wonderful presentation. Now the floor is open for any potential questions from the attendees. I already see some questions in the chat. Dr. Kastner, would you like to ask your question or speak about your comment?
Sure. Well, first of all, thank you. This was so insightful and packed with such important information. As an emergency nurse scientist, I'm really thrilled to see that BARDA focus on treat the injury, not the agent, and that agent agnostic approach. Kudos for that. I invite additional comments and perspectives on advancing this further in a meaningful field translation for a mass chemical exposure to treat the people in the population beyond one injury. MCM administration when there's concomitant trauma, blood loss, or in particular that social behavioral consideration we always have to deal with at the scene, whether that's that potential for crowd panic and stampede with crush, maybe vaccine hesitancy when we're talking about, an antitoxin or other vaccines, misinformation, and even violence targeting those first responders.
Yeah, I will just say I know that we are well aware of that, especially during the COVID pandemic, because there's a lot of that out there. I mean, I have family that actually did promote some of those misinformation, and so I was adamant to, well, you got to be careful with family, but so it is important to BARDA and to ASPR as well. We do have programs that are outside the medical countermeasure development that we are working on, trying to do that. We do work with social media trying to defeat that, but in the day and age, social media can be a problem. But we are working on it. We do have efforts like with trauma and blood loss. We do have blood products and other efforts, not within the ChemMCM, but it is within our burn blast. We also have Red Nuke that actually does look at, blood components and replacement. So there are different efforts out there besides that. What I've showed you, we also do a lot of our, I want to call, host therapeutic, host directed. So it's not just chemical symptoms. We do have, just like with ARDS or idiopathic pulmonary fibrosis, there are many different areas, like with viruses or with other biological effects that will be able to use or could use some of the same drugs that either we will piggyback on them or us. So we do, and even looking at all this, we do with our emergency response. That's one reason why we're also looking at needle-free capability with our auto injectors, because it is important. We also have different requirements in like the DoD where we do have to we make them have the retractable needles, because those safety aspects afterwards is also a problem. So hopefully I answered some of your questions, but I'm going to let other people step in too if they want to talk about them.
Yeah, thank you. Dr. George Anesi, do you have a question? I see that you raised your hand.
Yeah, thanks so much to both speakers. Fantastic information, very helpful, both from a research standpoint, from a funding standpoint, and kind of understanding what federal partners are looking at and how they function. Let's see, I have a bunch of questions, but maybe I'll start with all kind of preparedness research. I think all the way from basic science through translational science through kind of health services research preparedness can obviously is aimed at benefiting our preparedness, but can also expose vulnerabilities and be misused by kind of bad actors. I'm curious how your agencies approach the balance of research that, by the nature of being federally funded, I think often has to be public facing and transparent, with not contributing to kind of sensitive information that would be misused. Or how do you approach that kind of risk or balance?
Did you want to say anything, David? Do you want me to start?
Yeah. So that's that's a great question. That's something that I've definitely thought long and hard about, especially with how prevalent or how I guess in the spotlight, folks you having used AI ML to find therapeutics, finding in our world, finding potential analogs of certain chemicals. So yeah, yes, that's definitely a big area of concern for us as well. And you're exactly right, George. At least from the NIH side, everything we fund must be publishable. It's open to. So I other than that, I don't really have much I can say to get into mitigation strategies that are in place. And that is a big concern, especially when with the NIH coming to dual use research concern. That's a big push throughout the NIH, especially at NIAID, as everybody can imagine right now.
And it's similar for BARDA. We do have some, we have some stuff that have you need clearances for, and we do try to keep it secure. You'll see in quite a bit of our contracts, we do have your security for both personnel for the drug, your company itself, your IT, all that we do have that built in. So we are aware of that. And that's the thing. We're in the world now where as a researcher myself, you always have to worry about that, and that's the thing. It's a hard time balancing between your First Amendment and trying to get out your research, because it is important to where it can go back the other way, where they can use what you did to actually use it against you. So it's a fine balance. And I'm hoping as researchers and as a government, we are continually trying to improve upon that. But again, we also need that it's an ethical and moral issue too, so it's hard to control, but we're doing our best.
I have a question to Dr. Devon Andres. Dr. Andres, do you have a checklist that researchers can access and self-check if they have met all the mandatory milestones before they can reach out to BARDA?
I don't have one, because a lot of it is on a case-by-case basis. But I can kind of give you a little bit of one. As I said, you have to have capability of making your drug. You have to be able to have GMP manufacturing. It has to be able to be, because that's all part of your NDA approval. Even for IND, anything that the FDA needs, you have to have. Okay, so that's why we say talk to the FDA first. What is your strategy? You have to have a real world scenario on how to use it. In a lot of cases, like with the route of administration, we really would like to be able to use them, far forward as possible with less resource, for people to actually inject, do it. I'm not saying it quite right, but like auto injector is easy to use, one shot and you're done. Where if you were doing an IV for a long period of time, you're going to take up a bed, you're going to take up a person. So all those need to be considered. We can do IV and others, but we prefer other type of mechanisms that are easy and fast to deploy or administer. As I said, you need to have your clinical indication and have a lot of your toxicity studies in your preferably phase 1 already done on those clinical indications. I do know, like, if you haven't done anything on, say, your chemical aspect or efficacy studies, you got to remember, if you have a product even already approved for, say, something, if you're doing like want to do a label expansion or something, you got to remember the new formulation or a new route of administration or use, it's going to actually put you at an earlier TSL. So that's one thing you got to remember. If it's already approved and it's already up at a higher TSL and you do some change, it's back at an earlier one. So you got to kind of balance that. So we've had some people say, well, it was at a TSL 8. Well, you have no efficacy, no therapeutic strategy. You don't know what formulation you're going to need. You know, it does need to be changed because of the, bioavailability. So then again, is really at an earlier stage. So just remember, when you come to us, you're going to need all those things to make it successful. As I said, it is a, what do you want to say, case by case, because there's some we do take in earlier, but that's because of the type of compound candidate it is. Very novel. But then there's others that, it's been out there for a while, but you still need a little work, or has been out in the basic research for years and decades, but for some reason it hasn't come across. So those are, things to consider. You can always ask if you have questions on where yours are at. And one thing that is different between us and the DoD, I just got to remember, is like with the DoD, a lot of times, I don't know if you guys work with them, but we always get compared. With the DoD, they usually look at they have butt self and buddy aid, where the medics actually carry it there within their group. Well, in the public community, we don't have that. Okay, so we have to be able to have it successful one and done, or once you get to the hospital. We don't, well, and that's the other thing is it has to be done at, with an ambulance. The DoD, you get it first. One, and self and buddy, you can get it within 15 minutes because they're right there. Us, we have to wait a lot of times until the ambulance gets there, or you actually get to the hospital. So again, as far forward as we can, that's what we're looking for. And then we also have your special populations, like Dave Yeung mentioned. We have to look at our pediatrics. We have to look at those that are immunocompromised. Sometimes we can do get it through the FDA with the NDA approval, but then you have to do some work later to do those post-marketing, like with your pediatrics, or with, as I said, immunocompromised, like with your vaccines and stuff. So we need to know where your limitations are. You need to know where your limitations are and how to apply them, and how to get around them. I could probably spit out a lot more information, but it'd be nice if people actually had questions, because I could just keep ramble. So, because I have a lot of lessons learned, and I always want to help people if I can, with, provide them with information that they can use to be successful in their endeavors with their research. So, open to any questions.
Yeah, thank you. Dr. Shama, do you have a question? Please unmute yourself.
Yeah, so Dr. Andres, at what stage should a CCRP-funded investigator reach out to BARDA for the BARDA funding opportunities? And I was also wondering if there are any very successful products that received FDA approval for the support of CCRP and BARDA funding.
Okay, just to let you know, Dave actually showed some of it. It was with Acezalam. That started early on. We worked with JP on that and got IM formulation. So you get a lot of your basic research and translational data for applied, like he's talking about. There are some that was started off in CCRP, like the one I've been working on is Inversa. It is for ocular sulfur mustard injuries. We are currently getting ready to start our phase 3 trials for both AIK, or acute infectious conjunctivitis, as well as dry eye, and then starting now with the diabetic retinopathy. CCRP's program is starting to see more and more I'm not going to say it wasn't there before, but they're starting to do more research into the retinopathy that you see with sulfur muscle injuries. As I said, we do watch what our counterparts do, and we try to get them come over across as possible. Now, another one we have is the alteplase, the Genentech alteplase. Now, that is the one that I was really referring to when we start talking about animal rule versus, clinical indication with, not agent specific, because we're still trying to convince the FDA that, this inhalation model that we have, across multiple different animal models is the same as what we've seen in the humans. So, at the time, well, they didn't know that those casts in the lung were fibrin-based. And so, we got to show that, yes, those what they've seen back in the early 1900s, during World War I are the same as what we're seeing now in the animals. And so trying to do that is kind of hard. So all of it, as I said, we're close. So, but those are some of the takeaways that these programs are allowing us to give you lessons learned on. And so if you listen to, well, back to your first question, and when should you talk to us? And I will say, you can almost talk to us at any point. Now, I will say, if you already know, this is a little hard because each candidate is different. You should definitely be able to make it. Okay. If you're academia, I would say you definitely going to need to work with a company to be able to do the sponsorship a lot of times for the FDA, and for the manufacturing, mainly because a lot of times you don't have that experience that you need just in a small research lab. So you need to work with a company to be able to get it out. It's always important. If it's just you in the lab, it's hesitant because you got to be able to manufacture it. You got to market it. You got to do all those things that it's hard to do as a researcher. You do need, again, meet with the FDA. It's always important to do that. We can even answer some questions for you before you go to the FDA, but, up to end of the day, it's on you. So I would also say, know a lot of what your toxicity, have a lot of your toxicity studies, toxicity and safety studies done. That's also important. And if you're getting ready to go into phase 1, I guess the one thing too is, what is that clinical indication you're going for, and where are you with that as well? Because you can't just, as I said, we try not to do the agent anymore. Like with Cezolam and Midazolam, we are looking at for status epilepticus, anyhow, for seizures.
Exactly.
And so we don't even have nerve agent or organic phosphate on the label at all. So you have to know and have that clinical indication that you're going for beforehand. And so you need to do your research ahead of time as well. So, but we're willing to talk to you even as early as you want. We can't guarantee anything, but we can try to give you some advice as much as possible.
Thank you.
Dr. Yeung, it's so good to see that 70% of 2024 recipients of CCRP grant awardees are new investigators, at least to the CCRP program. That's a great. I see some, names here. They are new to our CCRP program or funding. So what is your piece of advice to these new funding seekers, or at least the new investigators to the CCRP program?
So for folks who are interested in applying, my advice is reach out to the program officer, talk to us about your science, and we can help guide you in terms of the right mechanism to apply to, because that's always critical. And because these funding mechanisms, some of them are reviewed by your normal standing study sections who may have different criteria threshold that they're looking for, versus other sections where it's special emphasis panels who are brought in ad hoc, and they're instructed to focus on something different. So, that I think that is the critical part in terms of folks looking to come into the program. For folks who are already in the program, who are brand new, the best advice I would give is for them to reach out and contact their fellow grantees in the program who have worked in with these chemicals in the past for a while now, who know more about the mechanism of toxicity, their potential therapeutic approaches, MOAs, and discuss and collaborate. I think that's the critical part, because that's what we want to push, is to build that scientific knowledge base and build collaboration, because no one lab is going to be able to have that magic approach.
Yeah, thank you. That's very, very helpful to the new funding seekers within the program. So are there any questions from the audience? Otherwise, in the interest of time, we can wrap up with a word of thanks. Okay, wonderful. Thank you again, Dr. Yeung and Dr. Andres, for taking time to present this wonderful webinar. I'm sure it will be very, very useful for all the audience. And last but not least, I would like to thank all the audience who stayed until now. Thank you all.
Thank
Thank
you.
you.
Thank you.